This is the second video in the unit on Mendelian genetics. In this video, we will learn how to complete a dihybrid cross using the rules of probability and how Mendel used dihybrid crosses to develop his principle of independent assortment. After completing his monohybrid crosses, Mendel was interested in exploring how two traits are inherited. He examined this by completing a variety of dihybrid crosses, crosses in which his parental generation were true breeding and differed in two different traits. The example we will use is plants that had yellow and wrinkled seeds or green and round seeds. All of the F1 plants from this cross were yellow and round, indicating that these are the dominant traits. If we write in genotypes of these plants, the yellow wrinkled would be homozygous dominant for the yellow allele, and homozygous recessive for the wrinkled allele, which we will designate as lowercase r. The green round plant would be homozygous recessive for the color allele, and homozygous dominant for the shape. The F1 generation would be heterozygous for each trait. Mendel's main question was, do the two traits always have to be inherited together? So will yellow seeds always be wrinkled and green always be round? Based on these F1 plants, it is clear that the two traits do not have to remain together. This is the principle of independent assortment, which states that the segregation of one set of alleles of a gene pair is independent of the segregation of another set of alleles of a different gene pair. We further observe this independent assortment when we look at the F2 generation, where the offspring were always observed in a phenotypic ratio of 9:3:3:1. This ratio is always seen in a dihybrid cross of two heterozygotes, as long as the genes are on separate chromosomes. By using a Punnett square, you can see where the expected 9:3:3:1 phenotypic ratio of the F2 generation comes from. The key for setting up a dihybrid cross is to remember that there are now two genes, and each will provide an allele to the gametes. This means that the yellow allele could be packaged with the round allele, giving a gamete that has uppercase Y, uppercase R. Or the yellow allele could have gotten packaged with the wrinkled allele, providing a gamete uppercase Y, lowercase r. Gametes could also form that have the lowercase y instead, packaged with your uppercase R, providing a gamete with the green allele and the round allele, or little y could have been packaged with little r, providing a gamete with the green allele and the wrinkled allele. Since the ovule gametes come from an individual with the same genotype as the pollen gametes, the possibilities for gametes will be the same along the top of the Punnett square. You will then fill in the boxes with each of the possible fertilization zygote combinations. You will notice as I do this that I'm putting the alleles for each gene together because it is easier to read the zygote phenotypes afterwards. Once all the boxes are completed, then we can examine the genotypes to determine the phenotype of each zygote. When we do this, you will find that there are 9 yellow round, 3 yellow wrinkled, 3 green round, and 1 green wrinkled. The principle of independent assortment holds true for genes located on different chromosomes. These are also called unlinked genes, and is based on the fact that when homologous chromosomes line up during metaphase 1 of meiosis, they can line up in either orientation. For example, if we have a cell that is heterozygous for the A and B genes, then when this cell enters meiosis 1, the homologous chromosomes with the A gene could line up so that the dominant A allele is on the same side of the cell as the dominant B allele on a non-homologous chromosome. If this occurs, then the resulting gametes would have the dominant A-B combination or recessive A-B combination. However, there's an equal probability that the dominant A allele lines up on the side of the cell with the recessive B allele. If this occurs, then the resulting gametes would contain the dominant A with the recessive B or a recessive A with a dominant B combination. Because the two orientations are equally likely, the overall ratio from a large number of cells undergoing meiosis is expected to be a 1 to 1 to 1 to 1. Dihybrid crosses can be more challenging and time-consuming than monohybrid crosses because they require a 16-box Punnett square and 4 gamete combinations. But consider if you were trying to do a trihybrid cross. This would have 8 possible gamete combinations and a 64-box Punnett square. This is not realistic to do, so geneticists use statistical probabilities and the forked line method to mathematically calculate the offspring. To do this, you must first understand the and or rules of probabilities. These rules explain how to combine different probabilities together. The and rule states that if the probabilities are independent events, which means you say and between them, you will multiply those probabilities together. For example, going back to our pea color crosses, if we cross 2 heterozygous yellow peas together, what is the chance that the pea pod, the seed closest to the stem, is green and the rest are yellow, looking like this? To calculate this, we look at the probabilities for each individual pea and then combine them. From a heterozygous cross, we can draw our Punnett square to determine what the likelihood is of each of the phenotypes. The Punnett square shows that there is a 1/4 chance of having a green pea and a 3/4 chance of having a yellow pea. Looking at the peas in the pod, we have a green pea and a yellow and another yellow and another yellow. Since we are stating and between them as we describe it, this tells us that they are independent events, and we will multiply the individual probabilities together to answer the question of what is the chance we get an entire pod with this color setup. When we do that, we see that the answer is 1/4 times 3/4 times 3/4 times 3/4, which gives us a 27 out of 256 chance of having a pea pod in this orientation. However, what if the question was, what is the probability that any seed was green and the others were yellow? This is not the same question we just answered, because the green pea could have been the first pea in the pod, or it could have been the second, or the third, or the fourth. Because we are saying or in between each possibility, we will calculate the individual probabilities, similarly how we did on the last problem, and then we will add the probabilities together. This is because the OR rule states that the probability of either 1 or 2 mutually exclusive events, i.e. one event or the other can occur, then we will add the probabilities together. Each individual pea pod has a 27 out of 256 chance of occurring. Thus, when we add them all together, we obtain a 42% chance that there is 1 green pea and 3 yellow peas in one pod. So how can the and or rules be used in a dihybrid cross? As long as the two traits being studied are on separate chromosomes, described as unlinked genes, then we can treat them as mutually exclusive events. The plants will be yellow and round. So you can do one monohybrid cross for the color gene and one monohybrid cross for the shape gene, and then combine the probabilities together using the AND rule with the forked line method. For example, going back to the cross of two heterozygous plants, instead of doing a 16-box Punnett square, we can do two separate monohybrid crosses, one for the color gene, where we see 3/4 yellow and 1/4 green, here's our 1/4 green, and one for the shape gene. Where we see 3/4 round and 1/4 wrinkled. We will then use a forked line method to combine these probabilities. To do the forked line method, you look at the probabilities of the first gene, and then draw forked lines from each of the probabilities to the second gene. You then combine each of the probabilities together based on the AND rule. For example, there is a 3/4 chance that the plants are yellow and a 3/4 chance that they are round. So there is a 3/4 times 3/4, or a 9/16 chance that they are yellow and round. Notice, as you go along each line, you will obtain the same 9:3:3:1 ratio that you obtained from the large Punnett square. It is also important to realize you do not always need to complete all branches of the forked line method, depending on what the question asks. In this example, it is only asking for the probability of one phenotype, so that's the only branch we need to worry about calculating. To complete the cross using probability calculations, we will look at the two traits as separate monohybrid crosses first, and fill out the Punnett squares. And then the second Punnett square, next, we will calculate the probability of just the offspring the question is asking for. The question wants to know what is the chance we will attain a plant with yellow, round peas. Looking at our first Punnett square, there's a 3/4 chance that we will attain a plant with yellow peas. Based on the second Punnett square, there is a 1/2 chance we will attain a plant with round peas. Since the peas will be yellow and round, we will multiply those probabilities together to obtain a 3/8 chance of having an offspring with yellow and round peas from this particular cross. A key to completing genetic problems is to be able to pull the required information out of the problem itself. You should remember that you will always be given enough information to work through the problem. However, oftentimes you will need to examine multiple generations to obtain all the necessary information. Here is an example of having to figure out an individual's genotype based on information provided. You should pause the video, read through the problem, and try to complete it on your own before watching my solution. As you read through the problem, it is always a good idea to write out key information. For example, the dominant allele P represents patches, while the recessive allele is solid. Notice that if the upper and lowercase letters look similar, I put a line under the lowercase letter to help you tell the difference. For the second gene, the dominant H allele is short hair, and the recessive is long hair. Next, we look at the cat phenotypes that are part of the problem. We have a long-haired cat with patches, whose mother was short hair and solid. The long-haired cat was mated to a short-haired solid-colored cat, whose mother had solid-colored long hair. For each cat, we will now fill in the parts of the genotype we know. Since this is a dihybrid, each cat should have 4 total alleles, 2 for the first gene and 2 for the second. For any traits we are initially unsure of, we will leave a blank until we can figure out the correct allele. Short hair was dominant to long, which means each short-haired cat must have at least one dominant H allele. But we do not know if they are heterozygous or homozygous dominant, so we will leave a blank for the second allele currently. This will be the case for each short-haired cat. This will also be the case for the dominant patches trait. Each cat with patches must have a dominant P, but we are unsure of the second allele because they may be heterozygous or homozygous. For the recessive traits, the cats must be homozygous recessive, or else we would not see the trait in the phenotype. So all long-haired cats must be little h, and all solid cats must be little p. All that remains is to determine if the dominant traits were heterozygous or homozygous. Looking at the short-haired solid mother, she had a kitten with long hair. This tells us the kitten had to get a recessive allele from both parents, so the mom must be heterozygous in order to have a recessive allele to pass to her offspring. Using the same logic, the offspring with patches must be heterozygous because mom only had recessive alleles to pass on. This cat must have gotten the dominant patches allele from the father. This pattern can be repeated with the other cats to fill in the remaining alleles and solve the problem.